of DNA6 for photoproduct. So Carlos, the stage, the screen is yours.
It's mine. Yeah, thank you. Can you see my screen?
Yes.
Okay. So I would like to first thank the organizer of the meeting, of the European Society for Photobiology meeting, and of course Virgin and Danielle for the kind invitation to present here in the photo damage symposium. I delete many of my introductory slides because I seen that Virgin and all the other speakers before me did a great job in presenting the background, why we care about DNA damage. And so I just gonna try to summarize more briefly here. Hopefully that will give us more time for discussion at the end. And so as everyone in this audience is well aware, sunlight is known to be correlated with skin cancer in humans, right? And in particular, as I'm showing here in this slide, there are three types of skin cancer that are of major concern. There are basal and squamous skin cancer. These are correlated to direct DNA damage by UVC and UVB radiation. But there are also very aggressive type of skin cancer called melanoma skin cancer, which are often associated with indirect UVA photochemistry. So UVA radiation exciting other biomolecules within the skin, and then those photosensitizing damage. Although there may be some evidence now more recently that UVA irradiation that can directly also damage DNA to some extent. And this is, of course, a major issue for society around the world. So skin cancer is one of the more prevalent cancer in the world. In the United States alone, there are more than 5 million diagnosed skin cancer every year. And this is $8.1 billion of health investment that is done to tackle this problem. So clearly, it's very important to understand DNA damage, and particularly what is the mechanism of the formation of these photoproducts. And I'm speaking to the quarry here. And so there have been a lot of work being done to understand and to try to obtain information about what are the main photoproducts that are formed and how that reaction happened. Because the hope is that understanding the mechanism through which DNA damage in the skin occur can lead to solutions of the problem, or finding by physicians, by dermatologists, way to avoid this type of reaction. Of course, as we hear Eva and in other sections, there is an enzymatic repair mechanism in the cells that repair 99% of this damage, but those that are not repaired can sustain long-term skin cancer. And so as we hear before by several of our speakers, the main photoproducts have, as well known by now in DNA, the particular the cysteine cyclobutane pyrimidine dimer and the 6,4 pyrimidine photoproducts are correlated with skin cancer. And so there have been a lot of interest in understanding the mechanism by which they are formed. The pyrimidine photodimer is from both, is the one that is formed in the highest yield. So almost 90% of direct photochemistry occurred in the formation between two thymine pyrimidine bases. And not surprisingly, the photochemistry and the reaction mechanism and the formation of this photoproduct have been studied extensively, and I will argue that is well understood now. And so I'm showing here compilations of research over the years where using femtosecond transient absorption, it has been shown that the reaction seems to occur in less than 1 picosecond, so it's very fast. And then using femtosecond transient IR, you can see the characteristic bands of the formation of the photodimer in less than 1 picosecond. So, so for what we understand nowadays, direct excitation of the photodimer, of the timing dimer from the single state produce this photodimer in sub 1 picosecond time scale. So this is a very efficient, very fast reaction. The reason for which is a low yield reaction is because it's also been shown by computational and quantum chemical calculations that it's very important the distance between the C6 and C5 double bonds and also the torsional angle. That conformation play a key role in the formation of the photodimer. And so the working idea is that because DNA is fluctuating in room temperature, now in the cell, it's not a static, it's not a fixed polymers that show here in this cartoon, is moving, then the particular torsional angles and distance between these bases are promoting conformational process in the 2+2 cycle addition reaction. So we have a significantly good understanding of this chemistry and how the reaction occur in double-stranded DNA. However, in the case of the 6,4 photoproduct, this has been more challenging. Okay, so we know that the 6,4 photoproduct is formed. There is no question about that. The analytical photochemists have characterized and have shown that this photoproduct is formed. There have been proposals that is formed through an intermediate, an oxetane intermediate, which have not been detected. So this is unknown. It's just a proposal based on the interaction of UV light and a reasonable mechanism is the formation of this oxetane intermediate. It's also not known what excited state is relevant in the formation. If it's from the single state, if it's happening from a triplet state, or if it happened from a charge transfer state, as we saw Roberto discussing earlier. And so there is all those unknowns. And also the rate, at what time scale this reaction occurred. So all those questions have not been very difficult to answer, even though there have been more than 60 years of continuous study of this process. Okay, so and for example, most of what we know about the reaction mechanism have fully been dependent on quantum chemical calculations. Okay, so again, the final product is well known, but what happened between, so this part here is what is not known, and we have a lot of information in the literature from theoretical chemists proposing different models to explain that reaction. And so I'm showing here some of them. So it have been proposed by different theoretical chemists that the M-pi single state could be the precursor state, the M-pi triplet state, the pi-pi triplet state, charge transfer state with both singlet and triplet character. So basically all the possibilities that you can think about have been proposed, and quantum chemistry from different level of theory support one model versus other. So all of them also predict the formation of this oxetane intermediate, which again have not been observed experimentally, and also the time scale in which is formed. So we have been thinking about how we can tackle this problem from the experimental point of view. I should say that, let me just go one more step back here. The problem of detecting these species using time result spectroscopy, so it's not all to blame to the theory, it's also experimental difficulties, is two reasons. First, is the final product is formed in very small yield. So you need to have a very sensitive instrumentation to detect the formation in time resolved process. But in addition, the absorption spectra of this transient species is postulated to absorb below 300. And so it's very difficult with the technical spectrometers that we commonly have available to be able to see this species time resolved. And so that's part of the reason why it has been so difficult to characterize. So now of what we are trying, we have tried to do. So what we thought was that we have been studying the photochemistry of thiobases now for more than 10 years. And I will not be talking about that today here, but in some of those experiments, we thought that we learned that the 4-thiothymidine, which is just a single atom substitution from an oxygen atom of thymine, not only absorb almost 100 nanometers to the red, but it also increased the yield of triplet state population by almost 85%. So this is a very highly reactive, highly efficient formation of the triplet state. And so we thought, and we can also selectively excite if we incorporate it in DNA because the absorption spectra is achieved. So we thought about the possibility of using this molecule as a potential model to study that reaction. Before I show you those results, let me give you a very quick background on the photochemistry of this 4-thiothymidine. This is transient absorption spectra, broadband transient absorption spectra from about 350 to 650 nanometers. What we have seen is the very fast population of the triplet state. This is a stimulated emission that occurs. So there is a fraction of the population that do not intersystem cross, indicated by fluorescent emission. And this band here in the deep UV is due to the ground state bleaching, so ground state depopulation. And so this was a very surprising result at that time because in organic molecules, intersystem crossing is usually spin forbidden and it takes a significant time. And so we observed a 240 femtosecond intersystem crossing. And so we wanted to understand that from a more quantum mechanical perspective. And for that, we team up with Inés Corral, that will be giving a short very shortly after me, and with Mauricio Persico to understand the quantum chemical dynamics using a multi-configuration interaction. The long story short, you can read it in the paper shown here, is that one of the key for this ultra-fast intersystem crossing is the observation of a three-state degeneracy regions that involve singlet and triplet state, where they are strongly coupled in such a way that intersystem crossing can happen very fast and efficient in these molecules. Okay, so having that knowledge, so we thought let's use this as a model compound. But there is another very important piece of information that I should mention, which is that the photochemistry of the 4-thiothymidine can be widely studied by Clivio and Fabre and others over the years. And very convenient for us, it has been shown by them that when you excite with UVA radiation, in this case at 360 nanometer, again, where the 4-thiothymidine base absorb uniquely, so you don't touch with photon energy the timing. What happened is that there is a very fast reaction that the time scale is not known, but until now, but they were able to show and actually to trap the Thietane intermediate, so the analog of the oxetane intermediate. This is the only intermediate species that have been isolated experimentally. And so they were able, as shown in this paper, they were able to show that this reaction occurred through this Thietane intermediate, and then that this rearranged to form the analog of the 6,4 photoproduct, where now instead of an oxygen, you have a sulfur atom. So not only this reaction, they were able to detect the Thietane intermediate, they also show in several papers, I'm just citing here the key one of those papers, that you can do these reactions in dinucleotides, you can do it in polymers, in single-stranded, in double-stranded, you get a very efficient formation of the 6,4 photoproduct, and equally important, without competition of the cyclopyrimidine butadimer formation. So the CPDs are not formed in this reaction. So we thought that this was, and the stereochemistry is well understood, which I will not discuss now. So we thought this is a great model to study the formation of 6,4 photodimer using a single-stranded oligonucleotide as a model of DNA. And so I'm showing here the steady state results initially. So this is a 7-mer oligonucleotide, which have 2 4-thiothymidine incorporated. The reason that we put 2 4-thiothymidine, we only need 1 to do the reaction, is because we wanted to have enough absorbent in the UVA to be able to selectively excite. You can imagine when you put one thiobase inside a polymer, you dilute the absorption or the concentration. So we have to use, that's just technical reasons. You can do the same reaction with only 1. But this is showing here the absorption, the normalized absorption spectra, which we are comparing with the monomer, with the 4-thiothymidine. You see that the only additional band shown here in the red, around 260, is that of the absorption spectra of the thymine bases. But other than that, you see just the characteristic absorption spectra of the 6,4 of the 4-thiothymidine, and slightly redshift when you put it in the single-stranded polymer. Here I'm also showing the emission spectra. This is all color-coded. And so for the monomer, you see 2 emissions band. We have showed that before, that this is due to fluorescent emission, while this significantly Stokes-shifted band is due to room temperature phosphorescence. So the triplet state is populated so high yield that we can even detect room temperature fluorescence in this molecule, and which can be quenched by oxygen and so on. In the case of the polymer, to our surprise, we only see fluorescent, and very small amount of fluorescent. This actually multiply just to this is normalized fluorescent, but not phosphorescent at all. So that suggests to us that as soon as you put the 4-thiothymidine inside a polymer, you quench a significant fraction of the triplet state, because we cannot see phosphorescent at room temperature. And this is even purging with nitrogen, removing oxygen from the solution. And further evidence that the triplet state is quenched was obtained by direct observation of singlet oxygen. So we have done it for the 4-thiothymidine monomer, and we observed a 40%, 42% singlet oxygen yield, just looking at the near IR emission of singlet oxygen. When you do the same experiment back-to-back with the polymer, you see now a 10-fold decrease in the singlet oxygen generation. So definitely a significant fraction of the triplet state is being quenched when you incorporate 4-thiothymidine in the oligonucleotide. And so to provide even additional evidence of that in this particular system that we are studying, we can form the 6,4 photodimer. We did irradiation experiment, irradiating at 342 wavelength, and this is the dose that we use. And this shows the absorption spectra before and after, and the different spectra now shown in blue here. The different spectra show a characteristic absorption band at 330, which is well known, as we thought from Regine in the previous seminar, that is related to the formation of the 6,4 photoproduct, in particular to the pyrimidone component of it. And not only that, if we now take the irradiate solution and excite at 330 nanometers and obtain the emission spectra, we obtain emission spectra that absorb, I'm sorry, here, that absorb 390 nanometers. And that is characteristic also of the emission spectra of the C64 photodimer. In addition, if we now sit at 390 and obtain the excitation spectra, which is shown here in red, this is exactly where the 6.4 photoproduct should absorb. So all these data taken together support the idea that we're forming the 6.4 analog of the Thietane photoadduct. And we did an additional experiment, which was we changed the pH because it's well known that there is an equilibrium between the Thietane intermediate and the final 6.4 photoproduct. And so if you change the pH, you can displace that equilibrium to the final 6.4. And that's what we see. We see that by increasing the pH, you see an increase in the 6.4 photodimer. So all these data provide, besides the data done before, analytical observation by Clivio and coworkers, that we actually are forming the 6.4 photoproduct in this single-stranded oligonucleotide. So then we went and said, well, we have now a system that we can study using transient absorption spectroscopy and follow the, oh, my time is, so I have to go a little faster.
Yes, you have
here.
4 minutes.
Okay, so I will go faster here. Just so this is back-to-back experiments of the monomer and the polymer. You can see that initially the first excitation is the same. So the mechanism of formation of the triplet state that we think that is the same. But as time progresses, as shown in the last 4 panel here, you see first in the polymer an isobestive point and observation of new transient band in the 400 region, which is not observed in the monomer. In addition, if we focus on that transient spectral region, you see the formation of a species as the triplet state decays, where in the monomer you don't see that. And so all this information suggests that we actually are detecting that transient species, and with more experimental and theoretical support that we have shown in this paper, we have characterized that to be the triplet state of the Thietane intermediate, which is formed, which then decay to the ground state, and in the ground state decay to form the 6.4 photodimer. Importantly, this reaction, intersystem crossing, occur in femtosecond timescale again, but the formation of this triplet Thietane intermediate occur in less than 3 picoseconds. So it's a very efficient reaction. And in combination with calculations done by Cui and coworkers, quantum chemical calculations, we have mapped up the full mechanism. I just want to summarize some things here. Again, when you excite with UVA, the 4-thiothymidine, it just intersystem cross to a triplet. From there, you can form the triplet state of the Thietane intermediate, and within a lifetime of 250 picoseconds, you have this triplet Thietane intermediate minimum, where this bond here is not spatially formed. And so according to the calculations of Cui, when this bond is formed, intersystem crossing back to the ground state occur, forming the Thietane in the ground state, which then rearrange to form the 6.4 photoproduct. And so in conclusion, and apologies for the excess time. So we now think that we provide indirect evidence or direct evidence for this system that the triplet state can initiate the photoreaction of the 6.4 photodimer, at least in this system, and suggests that it potentially can also participate in the formation of the oxetane intermediate. One final thing that I want to mention is that this, as have been shown by Virgin and Miranda before, this 6.4 photoproduct can act as a Trojan horse to do more DNA damage. And so understanding this reaction is very important. The final thing that I'm going to say is that our resource support the idea that the triplet state can participate in the formation of the 6,4 photodimer. It doesn't rule out that in other conformation, other systems, other singlet or short transfer state could also participate in the reaction. And given the time, I will stop there. I will thank the NSF for funding, the students that did the job in the lab, and all of you for listening and for your patience.
Okay, thank you, Carlos, for the